So my name is Bianca, and I wrote a literature review on Alzheimer's disease titled Alzheimer's Disease, What Do We Know About It and How Can We Tackle It? So to introduce Alzheimer's disease, though I covered a wide scope of research, I am mainly focused on the debate and uncertainty about its pathology and how treatment approaches work around this. So to start, Alzheimer's disease is an important area of study due to it being one of the leading causes of death in the senior population. So as the UN stated, the global population of citizens older than 60 in 2017 was over twice than what it was in 1980. And it's expected, and the number of citizens aged 80 years or over is expected to triple by 2050. So understandably, the correlated number of Alzheimer's disease cases would increase as well. And the World Health Organization's Global Health Observatory stated that as life expectancy is increasing, healthy life expectancy is not keeping up to speed. Healthy life expectancy describes the age that someone can live up to without disease or injury. So overall, all of these facts clearly suggest an increased prevalence of Alzheimer's disease. So the content that I discussed in my literature review was the history of the disease, the pathogenesis, treatment approaches, drug opportunities, and animal models. And then I talked about the correlation between COVID-19 and Alzheimer's to bring it into a modern context. So I will be discussing all of these in my presentation. So to start with the history of the disease, here it's illustrated on a timeline. So it can be traced all the way back to 2000 BC in ancient Egypt, when the ancient Egyptians recognized some form of memory loss and realized that memory declines with age, then taking us to the Middle Ages, where senile behavior was recognized but viewed as a punishment for sin due to the heavily theocratic society at the time. Then in 1797, dementia itself was finally accepted as a medical term, bringing us all the way to the 20th century, where a patient named August was presented to Alois Alzheimer's clinic with signs of delusion and memory impairment. He then used her brain for research upon her death in 1906, and his findings was what instigated further research into Alzheimer's disease and its pathogenesis, instituting the main hypotheses. So what were the symptoms that she displayed? So memory loss, she was forgetful, repeated questions constantly, she was delusional, had increased suspicion and began hallucinating. She was aggressive and just wasn't herself, having violent outbursts and tantrums and behavioral changes, and deteriorating language. Her conversation and writing skills deteriorated and she couldn't hold a conversation. So focusing more on the neuroscience and biology behind it, Alzheimer's found in his post-mortem analysis that the regions in her brain we now know are responsible for memory, language, and judgment and thinking had shrunk. She had a thin cerebral cortex, and she had extracellular senile amyloid beta plaques and intracellular neurofibrillary tangles made up of tau protein. So this brings us straight into the pathogenesis of the disease. So AD pathogenesis focuses on the two main hypotheses, amyloid cascade hypothesis and the tau hypothesis. But there are several that are the roots of heavy debate between researchers and exemplify the mystery of the disease itself. So the first ever approach explaining the molecular pathology of AD was the cholinergic hypothesis, which focuses on cholinergic neurons. And then going back to the two of the main histopathological hallmarks in AD, beta amyloid plaques and neurofibrillary tangles, you can see the amyloid cascade hypothesis and the tau hypothesis. So the cholinergic hypothesis revolves around damaged groups of cholinergic neurons which use the neurotransmitter acetylcholine, and decreased function of cholinergic neurons is present in normal aging, but there is increased neuronal cell loss in AD patients. So severe loss of cortical cholinergic innervation is consistently recorded in Alzheimer's. However, patients with mild cognitive impairment, which is where some form of cognitive decline is observed more than healthy aging, but less than serious dementia, or any early form of Alzheimer's, have only a loss of cholinergic function, but without any actual neurodegeneration. So then we that takes us into the amyloid hypothesis, which focuses on amyloid neuritic plaques caused by abnormalities with the amyloid precursor protein, APP. This protein is fragmented by enzymes into soluble amyloid precursor protein, SAPP, and amyloid beta peptide, which is what forms the plaques due to its tendency to aggregate. So mutations in this APP gene can either increase the amount of AB peptide or produce a longer and stickier form, again, increasing the amount of plaques. AD patients have decreased metabolic activity to remove amyloid beta, causing accumulation. In normal individuals, beta and gamma secretase extracts amyloid beta from APP and it immediately gets degraded when released outside of the cell. But under the pathological conditions of an Alzheimer's patient, individuals will have decreased metabolic activity to remove it, creating these plaques. The tau hypothesis focuses on neurofibrillary tangles made up of phosphorylated tau proteins. So the tau pathology and phosphorylation brings up a tense argument between scientists, so the amyloid versus the tau hypothesis. So microtubules are stabilized by microtubule-associated protein tau and are able to handle events like phosphorylation or other post-translational modifications. However, upon being hyperphosphorylated, tau detaches from these microtubules and forms NFTs and paired helical filaments, PHFs, as it accumulates. So zooming into the debate between the two main hypotheses, as some scientists would call it, Taoists versus Baptists, the amyloid hypothesis has many successful treatment opportunities centered around it, suggesting it must be somewhat accurate. So aducanumab has been proven to slow cognitive decline through inhibiting amyloid beta accumulation and has been FDA approved and in use since 2021. But the tau hypothesis is more closely correlated with the severity of Alzheimer's disease than amyloid plaques are, due to the processes such as microtubule assembly. And neurofibrillary tangles are also not seen in individuals not experiencing neurodegeneration, but amyloid plaques are seen in normal or healthy aging. The treatment approaches are all centered around the hypotheses, and understanding these is crucial when it comes to deciding how to overcome the disease, either by preventing it before onset or by delaying the progression or by curing the disease entirely. So for example, the cholinergic system damage can be pharmacologically tackled by using cholinergic intervention methods. So new drug development approaches for Alzheimer's disease that have arisen as a result of knowledge on the cholinergic hypothesis include acetylcholinesterase inhibitors, cholinergic precursors, cholinergic receptor agonists, and allosteric cholinergic receptor potentiators. But the main ones would be treatments targeting the amyloid hypothesis and the tau hypothesis, and a therapy approach known as integrated stress response. So treatment approaches targeting the amyloid hypothesis. So the beta secretase enzyme, BACE1, splits APP, releasing the N-terminus N from the amyloid beta peptide. So as this is significant in the generation of beta amyloid, inhibition of this proteolytic enzyme may decrease the cleavage, which is why it was explored more as a potential treatment opportunity. And then gamma secretase inhibitors are similar to beta secretase ones, except gamma secretase cleaves more proteins than just APP, making it harder to test, as some of these other proteins are important for healthy brain development. Antibody approaches have also been explored. So human amyloid peptide immunization of transgenic mice can invoke B cell and T cell responses, which is the model for antibody treatment. So in studies by Schenk and colleagues, significant amyloid deposition reductions were found in mutant human APP transgenic mice immunized with beta amyloid peptides. And other studies have shown as well that passive immunization with direct antibodies to beta amyloid penetrating the brain can reduce the deposition due to the antibody binding, potentially activating microglial mechanisms, which play an important role in brain inflammation. And then in regards to neuroinflammation, anti-inflammatory agents can also be used, such as non-steroidal anti-inflammatory drugs, which are also good treatment opportunities due to the beta amyloid receptors and receptors for advanced glycation end products interaction inducing inflammatory responses. So treatments targeting the tau hypothesis. So drug development opportunities tend to focus on targeting the mechanisms that convert the soluble monomeric tau proteins into insoluble tau aggregates made up of non-phosphorylated tau. Inhibiting this aggregation could slow the progression of Alzheimer's disease. Other drugs focus on inhibiting hyperphosphorylation, which is, like I mentioned, is what typically forms NFTs. So the activation of protein kinase activity is what is generally accepted as the cause of accelerated tau phosphorylation. And so protein kinases such as glycogen synthase kinase, cyclin-dependent kinase, and extracellular signal-related kinase are often targeted due to their correlation to the presence of NFTs. The integrated stress response is an intracellular signaling pathway in eukaryotic cells that uses the logistics of evolutionary conservation to help an organism or its cells and tissues to maintain health in a new environment. It reprograms gene expression to restore cellular homeostasis. And protein synthesis is reduced, but it also induces the translation of specific mRNAs, like the key transcription factor ATF4. And these processes all allow ISR to instill balance and reestablish homeostasis within the cell, yet there are other solutions, such as apoptosis, that ISR may resort to if stress cannot be alleviated. There is certain evidence to suggest that alleles such as the apolipoprotein allele can play a role, such as promoting key features of AD pathology like amyloid beta production and tau aggregation, though there's little research on the familial or genetic nature of Alzheimer's disease. So ISR elements such as protein kinase RNA activated have been shown to assist memory suppression in Alzheimer's disease, and so a recent study used a mouse model test of PKR could intervene with the harmful effects of ApoE4 on memory, concluding that ISR could potentially prevent related cognitive impairment, which is associated with AD. So then there are several drug opportunities focusing on each of these hypotheses. So there are cholinesterase inhibitors, again, focusing on the cholinergic hypothesis, and it essentially stops acetylcholine from being broken down in the brain. A monoclonal antibody, like I mentioned, aducanumab, targets amyloid beta plaques, reducing it and slowing cognitive decline. So both of these two drugs have been FDA approved, one in 2010, since the cholinergic hypothesis was one of the initial understandings of Alzheimer's disease, and one approved since 2021, the latest breakthrough in Alzheimer's disease. There are others in clinical trials at the moment. So nabilone, which is a synthetic cannabinoid, and it's an anti-emetic drug, reducing agitation through mimicking the euphoric effects of cannabinoids, and it's in phase 3 of clinical trials. Trisaprolin is a saprolic triglyceride, inducing ketosis and improving mitochondrial metabolism and neuronal function, which is in phase 2/3. And donanemab, another promising antibody targeting amyloid beta peptides, clearing the excess protein plaques, which is in phase 2. Animal models form the basis of drug testing and understanding Alzheimer's disease and testing any treatment approach. So transgenic mice are mice that have undergone a genetic intervention in their genome and are able to mimic AD in humans and demonstrate similar pathology when overproducing mutant APP. However, understandably, it cannot directly be compared to human pathology. So for example, APP overexpressing mouse models can show an increase in tau hyperphosphorylation, but they don't develop neurofibrillary tangles, making the amyloid hypothesis much easier to research on in mice, whereas the tau hypothesis is not. So the image on the left is an image that I included in my literature review, and it focuses on different studies on transgenic mice. So several APP transgenic mice models were successful in producing cognitive impairment that parallels what is experienced in a human patient. So for example, Games et al. Study using the human Indiana mutation on APP minigene as the transgene. It shows overall synaptic loss, neuronal loss in the hippocampus and dentate gyrus at 3 to 4 months of age, and general behavioral deficits in the mice. Bryan et al. Study using the human wild-type tau transgene showed hyperphosphorylated tau deposits and phosphorylation, but no neurofibrillary tangles. Like I mentioned, mice don't tend to usually show NFTs. Bullman et al. Study using both the APP23 with the Swedish mutation and the JNPL3 expressing the human mutant tau transgenes, showed accelerated tau pathology around the plaques, and Herzig et al. Study on APP Dutch gene crossed with the FAD mutant, familial Alzheimer's disease mutant, of PS1, creating APP PS1 mice, as well as Maya et al. Study on human APP crossed with PSEN1 to demonstrate familial Alzheimer's disease. So going to the correlation between Alzheimer's disease and COVID-19, recent research has shown links to show that this virus causing COVID-19 has the ability to invade the central nervous system, causing neurological symptoms to be seen as a potential comorbidity. The allele involved in the familial Alzheimer's disease might be involved in the pandemic as well, while also being the strongest risk gene for Alzheimer's disease. It's predicted to heighten the risk of a severe COVID-19 infection, which could increase mortality rates. So AD and COVID-19 also seem to have similar inflammatory responses due to an Alzheimer's disease patient's existing neuroinflammation, potentially influencing and speeding up cytokine elevation and therefore heighten the risk of mortality from a COVID-19 infection. Some social factors include that the fact that AD patients also tend to be unable to properly care for themselves and hence fail to follow social distancing guidelines or wear masks to protect themselves from the pandemic. So as a conclusion, Alzheimer's disease research has progressed greatly over time, as can be seen, and advanced scientific research into its pathogenesis has sparked the debate between the amyloid and the tau hypotheses. The frequent failure of clinical trials, however, suggests room for improvement within the scientific realm of neurodegeneration. And this can be due to several things. The presence of the debate itself exemplifies the importance of understanding such topics, but overall, due to the uncertainty surrounding the pathology, what aggregates first, what should be targeted for treatment, etc., treatment and other interventions should be comprehensive in their approaches to maximize chances of success. So for example, in the transgenic mice models that I showed, some included both tau pathology and amyloid pathology, which I would think is the most comprehensive approach to studying the disease, as it focuses on the two main histopathological hallmarks. But thank you.
Okay, well, that was a great presentation. Thank you so much. So for the last few minutes, I want to have the floor if any of them in the room has any questions or any comments. Yeah, I for one know someone in the room actually prepared some questions for you.
Hi, Bianca. That was a very interesting presentation. Thank you. That gave us a very good review of what's happening and a good intro of what's that. What do you think is the most important aspect of that to be dealt at the moment? I mean, do you think there is any specific scientific technology that needs to be improved, or any specific thing that you would love to see?
I think that the main issue right now is the fact that testing on mice can't be directly parallel to human pathology, and so it's hard to compare. And so when it comes to treatment approaches, it's hard to just use transgenic mice models to see what the best thing is. So that's why I think that being comprehensive with your approach and research, like including all the different hypotheses, as it's still the root of heavy debate between scientists, would be the best approach to finding not a cure, but any method of slowing down progression of the disease or prevention even. So I think that's what should be focused on in research.
Thank you. Thanks again for a very informative talk. Thank you.
Thank you.
All right, thank you. Do you have any other more questions in the room? Okay, great. Yeah, in that case, I want to thank you for coming to the symposium. That was a great presentation. Yeah, you can turn off the screen shares now. And now we'll be shortly expecting our next presenter. Thank you, Bianca.